Hope you had a good weekend. You guys studied a lot of biochemistry over the weekend? Yeah, right. Yeah, sure. You did? Good. Good? Kind of? Yeah. It's not sticking? Okay, well, hopefully it sticks. So put some glue on the biochemistry today. Okay, let's get started, folks. We've got a lot of material to cover, as you know. Shh. Thank you. Okay. Today I'm going to finish up, hopefully, although I'm not sure if I'll make it through it, talking about DNA replication. And I started talking about the subject on Friday. And as you're seeing, we have a lot of things to go through in this class. So that's one thing that happens a lot. All right. Well, let me remind you where I left you last time, which was right here. Okay. This figure is really, I think, central to understanding what the heck is going on. So there's a couple things I haven't told you, but I'm going to tell you, and that we will, they may not seem important. It may seem like just something that you memorize, but in fact, one of the things I'm going to tell you will help you to understand a lot of things better if you really learn it. Okay? One of the things I'm going to tell you is one of the few absolute rules in biochemistry. When I say absolute rules, it means there's no exceptions. In most things we think of, there's exceptions and so forth, but this one thing about which there's no exceptions is a very important thing, and that is that all DNA replication occurs only in the 5' to 3' direction. It only occurs in the 5' to 3' direction. That's what I showed you the other day when that new base came in and it got attached to that 3' hydroxyl. Okay, so when I talked about this polymerization right here, this is going from 5' to 3' because what's happening is each time I'm adding a new 3' base. All right? DNA replication only occurs in that direction. It will not go backwards. We can't make a strand going upwards. We can only make this strand going downwards. And that poses some interesting problems and issues for the process. Now, that comes back and hits us right in the face right here. Because remember that the strands are anti-parallel. The top strand, or my top finger, is going 5' to 3' from my left to my right, meaning your right to my left, or your right to the left, whatever, okay? And the bottom was in the opposite orientation. It's going 5' to 3' this direction, okay? When the replication fork is replicated, both strands are replicated at the same time. And this means that there are some hurdles that the DNA polymerase has to go through in order to make both of those processes happen at once. Because I can't simply reverse one process. I simply can't reverse that process because that would be making it 3' to 5'. They both have to go from 5' to 3'. And so what that means for all practical purposes is that the top strand is replicated, and then the bottom strand, and then the top, and then the bottom, and then the top, and then the bottom, and they're pointed in opposite directions. Okay? So let's talk about the top strand first. The top strand is the easy one to understand. The top strand is what we call the leading strand. The leading strand of DNA replication is a continuous strand, meaning that if I start over here, you see there's the 5' end, and you follow the purple is going as one continuous piece, and there's the 3' end right there. It doesn't get interrupted. It doesn't get pieces. It doesn't get put together in pieces, which is what happens to the other strand. Okay? It occurs as one contiguous strand. So leading strand replication is simple. 5' to 3', bang. The lagging strand is also 5' to 3', but because it can only replicate in the 5' to 3' direction, and the 5' to 3' is pointed in the opposite way, only a little bit of it can be replicated at any given time. All right? How does that work? Well, the leading strand goes along, the helicase unwinds strands, the leading strand says, okay, we're going to replicate up to here, and it gets up to here, and it looks at the other strand and says, oh, we haven't replicated in that other direction. And that other direction is not pointed forwards, that other direction is pointed backwards. Because remember, the strands are opposite. So the replication on that bottom strand can't go in the rightward direction, it's actually got to go in the leftward direction. So that means that the bottom strand can only replicate a little bit at a time. Only as new portions of the bottom strand get opened up can it start and go backwards. Okay? The bottom strand is what we call the lagging strand, and lagging strand replication occurs in pieces. Now, for a more advanced class, I would say you have to understand the how and the why there are those pieces. For a class that we're talking about here where we don't get to spend a lot of time on it, I don't think you have to know the where and the how of that. Okay? If you want to memorize that, that's fine. If you want to understand the where and how of that and you want to see me about that, I'd be happy to tell you more about it. Okay? But suffice it to say, the leading strand is continuous, the lagging strand occurs in pieces. Now, another thing I want to tell you about this, and I mentioned it the other day, and that is that when we look at DNA replication, there are no DNA polymerases that will start replication all by themselves. There are no polymerases that will start replication all by themselves, meaning they have to have something start a strand before they can hop onto it. Okay? It turns out that there is an enzyme that will start replication for a DNA polymerase. And it's actually an interesting enzyme. It's an enzyme called primase, P-R-I-M-A-S-E. Primase is not a DNA polymerase because no DNA polymerases can start a strand. Well, then how does it get started? Well, it turns out that primase is an RNA polymerase. Primase starts DNA replication by putting a piece of RNA down. It makes a little piece of RNA. And then the DNA polymerase grabs that piece of RNA and it says, oh, here's a start. I'm going to extend from this point forwards. And so the DNA polymerase attaches to the RNA, and everything is fine. So a DNA polymerase is extending an existing strand. That's what a DNA polymerase does. It extends an existing strand, and that existing strand can be a piece of RNA. That existing strand can be a piece of DNA. Okay, so what does that mean? Well, it means that when we look at all of these pieces that are being made on the lagging strand, every one of those pieces starts out with a little stretch of RNA, followed by DNA, and then it gets to the next piece, which has RNA followed by DNA, RNA followed by DNA, RNA followed by DNA, and you get the idea that that lagging strand is really a mesh of a bunch of things. RNA, DNA, RNA, DNA, RNA, DNA, okay? It's because of this mix of things that's there that it takes 2 DNA polymerases to make it be finished. So I've been talking so far about one of the DNA polymerases. The one I've been talking about so far that's doing all the work that I've talked about so far is called DNA polymerase III. DNA polymerase III is the one you see right there, copying both strands. That's DNA polymerase III right there. The other DNA polymerase that does some work is called DNA polymerase I. And DNA polymerase I's job is really interesting. DNA polymerase I has to remove those little pieces of RNA and replace them with DNA. That's what polymerase I does. It removes those RNAs and replaces them with DNAs. So when polymerase I is done doing its thing, we've gone from RNA, DNA, RNA, DNA, RNA, DNA to something that looks like DNA, gap, DNA, gap, DNA. Because the RNA pieces have been removed, and the only gap is the fact that we haven't linked together those pieces. We have to tie those gaps together, and we tie those gaps together with an enzyme called DNA ligase. DNA ligase joins the gaps. It joins fragments of DNA together covalently. DNA ligase will create phosphodiester bonds. That's how it's joining those pieces together. Now, at the end of the action of polymerase I and DNA ligase, and you see they're linked very close together, at the end of that, the lagging strand is a single piece, just like the leading strand is a single piece. So the lagging strand starts out in pieces, but ultimately, in the very end, it all gets tied together. So that our double-stranded DNA doesn't have any gaps in it, doesn't have anything else in it. Everybody understand that? Yes, Rachel. An RNA starts for each of the little fragments. That also is what happens at the very beginning of the leading strand as well. So the leading strand starts out as an RNA fragment as well. And that has to be removed as well. You guys get that? That's either I'm getting better at explaining things, or you guys are getting more bashful about asking things, or you're asleep. Which one is it? If you're asleep, I'll take that as no answer. So that means if you don't say anything to that, then I'll assume you're asleep. You must be asleep, right? Wish you well, I can put you to sleep, I'm sure. Okay. Well, now you've seen the action of most of those proteins that are important at the replication fork. Okay? There's two that I haven't mentioned, and I'm going to... Oh, by the way, the fragments that we see on the lagging strand have a name. They're called Okazaki fragments. Okazaki fragments. That's what those pieces of RNA, DNA, RNA, DNA, RNA, DNA, that's what those fragments are called. Okazaki fragments. Okay. Now, I said there's two proteins that I haven't talked about. One of the proteins is up here labeled SSB. And SSB, I mentioned it the other day. I'll tell you what it stands for here. It's called Single Strand Binding Protein. Single Strand Binding Protein. In fact, if you look at this figure, you can see those little green circles, and you see what they're coding are single strands of DNA. One of the things that they're doing there is they're actually providing some protection for that single strand. If that single strand were to get damaged, the whole DNA would be useless. So one of the functions of the single-strand binding protein is to provide some protection for that single strand. It turns out it also plays a role in the replication process. It helps the polymerase, but we don't need to worry about that. Okay? But wherever we see a replication fork, we will always see SSB. The other protein that's on here is called the beta, what's labeled on here is the beta subunit. You see in parentheses, sliding clamp. Okay? The sliding clamp has a very cool and very easy to understand function. Okay? I told you earlier that DNA polymerases tend to fall off. If you put a DNA polymerase at a replication fork, it doesn't matter what the DNA polymerase is. If you put a DNA polymerase by itself at a replication fork, it will replicate for a little bit, and then it will fall off. Okay? Now, when that happens, okay, replication doesn't proceed very far. And it turns out that when we look at the two polymerases, one of the polymerases is very good at falling off, and that turns out to be very useful. Could you predict for me which one would be useful for it to fall off? Polymerase 1. Because it only goes for a little bit, it's finished its job, and it falls off. And it goes for a little bit, and it finishes job, and it falls off. Polymerase 1 is set up to fall off very nicely. Polymerase 3, on the other hand, this is the guy that does most of the replication of the DNA in a cell. It's the one that's going at 1,000 nucleotides a second. It's the one that we want in the trenches doing its thing. We don't want it falling off. And it's this protein that's attached to what we call the beta clamp. The beta clamp has a very simple function. First of all, the beta clamp binds to polymerase 3. The beta clamp binds to polymerase 3. The second thing that the beta clamp does is the following. The beta clamp looks like this, and it does this. Did you see what I did? It looks like this, and it does this. Imagine that I've got a strand of DNA, and now the beta clamp does this. What's going to happen? It can't fall off. The beta clamp is holding on to the DNA so that the polymerase 3 does not fall off. It holds the polymerase 3 at the replication fork. Now you'll notice I'm saying we. This is actually happening in E. coli. But what's happening in our cells is essentially the same. The proteins have some different names. We're not going to worry about those names for the most part. But the functions are the same. We have something almost identical to the beta clamp that holds the polymerase at the replication fork in our cells. Okay? Yes? So, the question is, does the beta clamp hold the DNA or the polymerase? The beta clamp is going around the DNA, so it's holding the polymerase to the DNA. Okay? So, because the beta clamp is there, you can see it wrapped around the DNA. The polymerase, which is attached to it, can't go flying away. The polymerase binds to the beta clamp. I'm sorry? The polymerase is the brownish part. That's right. Okay? So that function turns out to be really useful for holding the polymerase to the DNA. And that's why polymerase 3 stays on the DNA and polymerase 1 does not stay on the DNA. Polymerase 1 does not bind to the beta clamp. Okay. All right. You guys are in good shape. Now, that's the hard part of today's lecture, actually. Most of the rest of the stuff in today's lecture are pretty cool. Some of it relating to aging and cancer that we're going to talk about. Okay? But before we get to that exciting stuff, I've got to tell you something about the polymerases. Our cells and E. coli's cells have many DNA polymerases, many different DNA polymerases. Okay? In E. coli, there are actually 5 DNA polymerases, 3 of which have numbers and 3 of which you see on the screen. You've seen 2 of them so far. You saw polymerase 3 and you saw polymerase 1. Polymerase II is a relatively minor polymerase, and it's there to help the E. coli cell deal with damaged DNA. It helps the E. coli cell to deal with damaged DNA. For our purposes, we're not going to care about polymerase II. Polymerase 1 and 3 are going to be the ones that we will focus on. Polymerase 2, we don't care that much about. I want you to focus your attention, okay, on the bottom set of activities here. Okay? Activities. What are activities? Activities are things being catalyzed. All right? If we look at polymerase 1, we see that there's something that says polymerization 5 to 3, something that says exonuclease 3' to 5', and something that says exonuclease 5' to 3'. Okay? If we compare that to polymerase 3, we see polymerization 5' to 3', we see exonuclease 3' to 5', but we see no for exonuclease 5' to 3'. I'm going to tell you what those things are. These are all reactions that these polymerases will catalyze. Now, first of them we've talked about. The first of them was it makes DNA. That's polymerization. And the polymerization, you know, occurs in the 5' to 3' direction. So no surprise, both polymerase 1, polymerase 2, and polymerase 3 polymerize DNA. We wouldn't call them a polymerase if they didn't do that. Okay? The second function is really interesting. An exonuclease. What the heck is an exonuclease? Here's the way I like to describe it. Let's imagine that over the top of the class, I have a strand of DNA. Okay? Over the top of the class, I have a strand of DNA. And just beneath that strand of DNA, I have a DNA polymerase that has started copying an existing strand. So I got the top strand, and now it's the bottom one is copying, the polymerase on the bottom is copying the top one. Everybody with me? So the polymerase is going along, and it looks at the top strand, and it sees a G, and it puts in a C. And it sees a T, and it puts in an A. And it sees a C, and it puts in a G, and it sees a T, and it puts in an A, and it sees an A, and it puts in a T, and it sees a G, and it puts in a G. Uh-oh. I made a mistake. I put a G across from a G. I told you that polymerases make rare mistakes, right? If you are the editor of a journal, one of your jobs is to read the articles for the content, but the other function that you have as the editor of the journal is to look for typographical errors, because typists make errors. And you see somebody misspelled a word, oh man, I'm going to flag this, they got to fix this word, right? You are functioning in that capacity as a proofreader. You're proofreading and making sure that there are no embarrassing errors in the things that you're publishing, right? The polymerase comes along, and it's copying, it's putting the right things down, and it has now put a G across from a G. There is an embarrassing error, known as a mutation. Right? Well, if the polymerase didn't have the ability to proofread, it would make more errors. But this typist, which is our DNA polymerase, I told you how fast it's going, 1,000 nucleotides a second, it's proofreading as it's going along. So what happens is, here's this GG that's sitting here, okay? And the polymerase goes, oh, wait a minute, what's going on? And it stops. So the polymerase recognizes that it's made a mistake, it's put something in that it shouldn't have put in, and it stops. And when it stops, that G that shouldn't be there pops into a different site in the polymerase. We talked about the active site. This polymerase has 3 active sites. One where it makes the DNA. But now we've popped this into a different site, and the different site is where it's called this 3' to 5' exonuclease. And what the 3' to 5' exonuclease does is it breaks phosphodiester bonds. It starts at the end of a fragment, and it starts moving backwards, and it chops out nucleotides one at a time, and it does that by breaking phosphodiester bonds. So when I've got this GG, the wrong G now is there, pops into this other place on the enzyme, and the enzyme says, oh, I'm cutting you out. And so the enzyme, instead of moving forwards in the 5' to 3' direction, now starts sliding backwards in the 3' to 5' direction, chopping out a nucleotide, chopping out a nucleotide, chopping out a nucleotide, chopping out a nucleotide. It's chopping out some of those correctly done nucleotides. But most importantly, it's chopped out the mismatched nucleotide. Okay? Oop. Sure. Okay? So the proofreading has worked. The proofreading has said, this is not matched properly, we're going to remove it. And now the polymerase takes over and it goes back and it starts filling in nucleotides again in the correct direction with the correct base pairing. Everybody with me? Yes? Is there a set number of numbers that it cuts back? No, but it'll go back a ways. It won't just take out that one. It'll take out a few. Yeah.
So it goes forward first, and then it
Sees an error, it goes backwards.
and then does it refill it?
And then it'll refill it. So it'll go backwards for a little ways, and then it's got the error out, and then it goes back forwards again. Okay? Yes? How does it see it? That's actually a bigger question. It's a good question. But it turns out that, let's say the GG. G is a purine, and G is a purine. They're both big. You could imagine that would bulge. And it's actually the bulge that's the signal that there's a problem. And you might say, well, does it only work with GG? No, it doesn't. That's the interesting thing. Because smaller bulges happen if you don't form hydrogen bonds. So if you try to put a G across from a T, that would be a purine with a pyrimidine, you still get a little bit of a bulge because they don't have hydrogen bonds. It's a very cool question. It's a very cool problem. Okay? Did that answer your question? More or less. Other questions? Okay, I promise we'll get to some cool stuff, but I've got to get through this first. All right. The one other thing we've got to get through is what the hell is the 5' to 3'? An exonuclease, you've already learned, is something that starts at the end of a DNA, and it starts chopping nucleotides away, breaking phosphodiester bonds. But this would seem counterproductive. Why in the world is it starting at a 5' end and doing that? Well, we'll notice, first of all, that DNA polymerase III does not do this 5' to 3' exonuclease activity. Only DNA polymerase I has a yes there. Only DNA polymerase I is able to start at the 5' end of a nucleic acid and start chewing away nucleotides. Can anybody think of a reason why polymerase I might have that activity and polymerase III might not have that activity? What's that? Not because it falls off. What does polymerase I work on that polymerase III does not work on? The lagging strand. And what is on the lagging strand? RNA. The polymerase I has got to get rid of that RNA. So it comes along at the 5' end where that RNA starts, and it says, oh, chop, 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 and it chops out that RNA starting at the 5' end of the RNA, and behind it is a DNA fragment. It extends the DNA and fills that gap. And so that's how all those gaps get filled in there, is the 5' to 3' exonuclease chops out the RNA at the same time as the polymerase is filling it in with DNA. That's a pretty cool process. Okay. That RNA piece has a name. We call the RNA piece a primer. It's called a primer. That's why the enzyme is called a primase. It's called a primer. And we are getting rid of that primer and replacing it with DNA. Ultimately, all of those RNA pieces will get removed by polymerase I. Everybody with me? If I said, what's the proofreading activity in a DNA polymerase, what would you tell me? Define it. I would hope you would say it's a 3' to 5' exonuclease, right? If I said, what's the primer removing activity, I would hope you would tell me it's the 5' to 3' exonuclease activity, right? Which of these makes phosphodiester bonds and which of these breaks phosphodiester bonds? The polymerase activity makes phosphodiester bonds. The exonuclease activities break phosphodiester bonds. Okay? Okay. There's the beta clamp I told you about earlier. You see how it wraps around the DNA? The DNA is in the middle, and there's that circle it makes around the DNA. Kind of cool. Kind of cool. Okay. There's a whole bunch of stuff to not memorize. How about that? Okay. There's that same view of the beta clamp from the side. And let's go back to the replication fork briefly, and then I'll tell you why this is relevant. Okay? So at the replication fork, I want to reiterate two proteins that are ahead of the replication fork. Right? One, I've told you, was the helicase, and the helicase was important because it had to unwind and unwrap the strands to stay ahead of the DNA polymerase that's racing through there, right? So the helicase was the protein that I told you had to work at 6,000 RPM. This figure's unfortunate in that it shows the helicase right here, and it shows DNA gyrase as if it's immediately next to it, and that's not the case. It's exaggerated. In fact, the helicase is up ahead of things a little ways. I'm sorry, the gyrase. Sorry, I said helicase. The gyrase is ahead of things. The helicase is much closer to the replication fork. The gyrase is the enzyme that I told you was relieving the tension that was being created by the helicase as it unwound the strands. As the helicase unwinds the strands, ahead of it there's tension, and if we don't relieve that tension, we'll end up with a knot, and that tension is relieved by this enzyme called DNA gyrase. I told you that gyrase was a topoisomerase. And because it affects the tension, topoisomerases can affect supercoiling. Turns out topoisomerases can relieve tension, and some of them can create tension. They can work both ways. What you see on the screen moving from left to right is a topoisomerase that is increasing the tension. Okay. You can see it takes energy to do that. There's ATP being used and AMP that's being produced. Okay. All right. There's the proofreading. And there's that mismatched base that we saw before, I told you about. Okay. By the way, proofreading is really important for a polymerase. With proofreading, you improve the accuracy of a polymerase by between about 100-fold and 1,000-fold, meaning it's that much more accurate. I told you that E. coli's replication system made about one error in every 10 million bases. And it makes so few errors because it has proofreading. It makes so few errors because it has proofreading. If the polymerase did not have that proofreading, it would make about an error every 100,000 or so bases. Still really great, but not as great as 1 in every 10 million. And just like a person who's proofreading a paper doesn't catch every error, so too does the proofreader on the polymerase not catch every error. That's why you still see 1 error in every 10 million. But that's pretty good. Now, I tell you this because there are some DNA polymerases that do not have proofreading activity. They do not have proofreading activity. And as a consequence, they make errors much more frequently. The best known of these enzymes is the enzyme that makes the DNA for HIV. HIV's DNA polymerase does not have the ability to proofread DNA. It makes an error about 1 in every 10,000 to every 100,000 bases. That means that HIV's DNA polymerase, when it makes DNA, is much more frequently making errors than the cell's polymerase. And that means HIV is rapidly mutating compared to everything else. And you know that because HIV, over time, will escape drugs. That is, a drug that's effective against HIV today may not be effective against HIV tomorrow. And the reason is because HIV is always mutating. It's always trying to find a way around that drug, and it eventually finds one. Mutation is the way around a drug. Viruses commonly have enzymes that lack proofreading. Yes? Could that work against the virus's success? The answer is basically no. It only works for it because those that aren't functional no longer play a role. That is, if the virus has a mutation such that it's something that the virus needs, that one mutated virus won't function, right? But it'll never appear in the whole scheme of things. So it doesn't work against it as a whole. It actually works for it because it allows a virus many more possibilities and many more ways to infect things. So viruses will tend to mutate much more readily than cellular DNAs will. And HIV is a type of virus called a retrovirus, and of all the viruses that mutate, that's probably the most frequently mutating viruses that you will see, retroviruses. Okay. All right. Now, I'm going to talk about DNA repair in a little bit, but I want to say something about eukaryotic DNA replication first, because I think it's more interesting. Okay? Oh, man, we're not going to memorize all that stuff. Okay? There's a good piece of news for you, all right? We are going to think about a couple of things about eukaryotic DNA replication, but we're not going to go through all the names and all the blah, blah, blah of that. That would not be useful. That would not be a productive use of your time. Okay? Prokaryotic replication differs from eukaryotic replication in a few important ways. I told you earlier that prokaryotic cells have circular DNAs. Circular DNAs. Those circular DNAs replicate. They start at one place, and there's one replication origin on a prokaryotic DNA. One origin on a prokaryotic DNA. At that replication origin, replication forks. One goes this way, one goes this way, and they go up here and they meet at the top. Okay? One origin, two replication forks. They run into each other at the top, and everybody's done. You've replicated a bacterial DNA. That's not the case for eukaryotes. First of all, eukaryotic chromosomes are very long. Hundreds of times longer, thousands of times longer than that one circular DNA. Consequently, eukaryotic chromosomes have multiple replication origins. They don't have one. They have multiple. But at each replication origin, we get a fork, just like we had in prokaryotes. We have single-stranded binding protein, just like we had in prokaryotes. We have primase, just like we had in prokaryotes. And they have their own names, but we don't care about the names here. The basic setup is the same, but there's one place where there's a very important difference. And the place where there's a very important difference is when we get to the ends of the linear DNAs. At the ends of eukaryotic chromosomes, there's something that's really interesting there. Now, first of all, I'm going to tell you the problem, and then I'm going to tell you what's interesting. Here's the problem. What did I tell you about DNA polymerase's ability to start making a strand? It can't do it, right? No DNA polymerase can start making a strand by itself. You saw that it took primase to make that happen, right? And what was primase making? It's making a little piece of RNA, right? Let's imagine that we're at the end of a chromosome, right? And at the end of a chromosome, we have a primase that starts at the very end, and it makes a little piece of RNA to start that replication. You with me? DNA polymerase says, oh, there's a start. I'm going to extend it and move and do my thing. In the case of a circular DNA, right, the polymerase comes back around, the polymerase 1 comes back around, and it now runs into that RNA, and what does it do? It removes it and replaces it with a DNA. With a linear DNA, it can't do that, because that would mean it would be starting a DNA at the end. And it can't do it. Consequently, every time a eukaryotic linear DNA is replicated, there's a little piece of DNA at the end that is lost every time replication proceeds. Every time replication proceeds, a little piece of that DNA is lost at the end of a linear eukaryotic chromosome. For all practical purposes, what does that mean? It means that if you were to examine my chromosomes and compared them to your chromosomes, and we looked at the ends of my chromosomes, what we would discover is that my chromosomes are shorter than yours are. My chromosomes are shorter than yours are. When it comes to chromosomes, folks, size matters. Size matters. Imagine that the longer and longer and longer you replicate a cell, the more and more and more of the DNA you lose. Do you see a problem? I see a problem. Because I've got genes on those chromosomes that are important. And if I start losing, at some point I'm going to lose some gene that I need for that cell, right? One of the models people have of aging is that with time, you're losing chromosomes, you're losing functional genes that you have to have, and that's part of what aging is about. Well, there's a couple things that help to bail us out. A couple of things. All right? Well, what are they? One of them is that at the end of our chromosomes, we have an unusual sequence. We're born with it. It's called a telomere. T-E-L-O-M-E-R-E. What is a telomere? I'm going to define it for you. A telomere is a sequence at the end, or at each end, of a linear eukaryotic chromosome. And it consists of the following things: thousands and thousands of repeats of the same sequence, over and over and over and over. That sequence is about 7 to 10 base pairs long. Depends a little bit on the species, but that's about 7 to 10 base pairs long. And it's repeated thousands of times. Now, based on what I just told you, I hope you can start to see the function of the telomere. The telomere doesn't code for any genes. It's part of what some people would call junk DNA, because things that don't code for genes, people call junk. It isn't completely true. For me, I think of it as a fuse. If I'm going to go light a stick of dynamite, right? And I want to get away from it, I want to have a nice long fuse, right? So I got a stick of dynamite sitting over here, and that sucker is going to blow, and it's going to cause a big boom a long ways away. I want to get enough time to run away from where the big boom is going to go, so I take my match, I light the stick, and I go running to beat all hell. The longer that fuse is, the more likely I won't be affected by that boom going off, right? Everybody understand? The telomere is a fuse. As long as I'm in the fuse region, I'm not going to get blown up. Once I start going past the telomere region, guess what's going to happen? I got some problems, don't I? All right? So I want to be born with long telomeres. The longer my fuse is, the more divisions I get, right? That's what I want. How do I get those telomeres put on there in the first place? I get them put on there by an enzyme. When I'm a fertilized egg, there's an enzyme that comes along, and it's called a telomerase. T-E-L-O-M-E-R-A-S-E. A telomerase's job, and I'm not going to make you responsible for knowing how it works. I'm going to tell you some simple details about it, but I'm not going to hold you responsible for knowing how it works. But a telomerase's job is to make telomeres. Thousands and thousands and thousands of copies of the same sequence over and over. It makes that because it carries with it the sequence that it copies. It carries its own template to copy. And that sequence is about 7 to 10 bases long. So it copies it once, it copies it again, it copies it again, it copies it again. It does that a few thousand times. And when it's done, I have this long telomere at the end. Some people think that the longer your telomeres are, the longer you live. And based on what I've told you, you might think that's true. And there's some evidence, at least weak evidence, that there's some truth in that. So you start saying, I want my telomeres to be as long as possible. How come my telomeres shorten if I've got a telomerase? And they shorten because telomerase, unfortunately, is only found in embryonic cells. Well, there's a couple of other places it's found too. It's found in stem cells, which are capable of regenerating and doing things. Okay? And some people say, well, if we want to live forever, we should make telomerase active. Can we make it active in our cells? Was that your question? No. If a baby is born prematurely, does it affect their telomeres? Probably not. Probably it's all established by then. Good question, though. Okay? If I make my telomerase active, will I live forever? Well, yeah, we have cells that do that. They're known as cancer cells. They will live forever, and they live forever because they have a telomerase that keeps replacing what's there. We want to be careful before we make these completely active, because if we're not careful, we create a cancer that we can't control. Okay? One of the strategies for killing a cancer is to kill the telomerase. Because now the cancer is going to burn itself out with all the divisions that it's doing. Okay, I know I'm running on just a little bit here, but this is an important topic, okay? Questions about that before I do a song? Yes?
The copies that the telomerase makes, they hear their own template, but is it the same exact one for all of them, or does it match up with the exact?
So it's one template, and it's copied thousands of times, the same one, over and over and over. It's not. It's just a template is all it is. It's just copied. Yeah. Okay. This is a fun song. I hope you enjoy it. It's called the 3 R's of DNA. Yep. I thought it was. Hold on. Oh, I know what it is. Hold on. Shh.
Fix the DNA for me. Such pathways of excision cause cells to have to make a decision. Should they go straight ahead with repair or take themselves right out of there? Then lastly, there's recombination. Swap strands readily. Crossover molecules, regions. Mix them for me. This story is complete now. The DNA is fit for gametes now. The 3 R's for the DNA shine. Replicate, repair, recombine. Oh yeah. Replicate, repair, recombine.
All right, guys. See you tomorrow.